Hello to our friends joining us via recording. Today we are wrapping up lesson number 12. To help us wrap up lesson number 12, we're going to start with a review of what we talked about last time. Remember that one of the things we talked about during last week's class was reflexes. We talked about how reflexes are things that happen inside the gray matter of the spinal cord. When we talk about reflexes, I promised you that on the exam, you're going to get a picture of a reflex that you've never seen before. You won't know its name, you won't know anything about it, you'll just have the picture. And I'm going to ask you to classify it just like we see down here with these kinds of words. So this is a picture of the reflex model from lab. We're going to use this picture to help us make some predictions. When we look at this picture of a reflex here, notice that our reflex is starting out here at the skin. If you remember from lab, we talked about how these little green spots are kind of these areas here on my sensory neuron out here. These are our receptors where we're detecting information. We don't know for sure what kind of information this reflex is detecting. I would suspect it's probably something like temperature, for example. So something your skin detects. That sensory information is coming into the spinal cord. Remember that my sensory neurons are unipolar neurons, so there's a classification word for us. They live back here in the dorsal root ganglion. This sensory neuron is bringing its temperature information into the spinal cord, where my interneuron, who processes that information, where they live, they figure out how we should be responding and send that message out via a motor neuron that goes out to, in this case, a skeletal muscle to make the body move. So we're looking at a very typical reflex arc. In this reflex arc, we had a sensory neuron, and then an interneuron, and then a motor neuron. We had 3 neurons in this reflex arc. Does that tell me that this one is monosynaptic or polysynaptic? If I have 3 different neurons in my reflex arc. Yeah, exactly. So this is going to be an example of a polysynaptic reflex. The word polysynaptic, this synaptic part of these words, tells me the number of synapses, So I have some reflexes that are monosynaptic. I have 1 synapse, but most of my reflexes in the body are polysynaptic. I have more than 1 synapse or more than 1 place where my neurons meet. So if we go back up to our reflex arc that we see here, notice how we have synapse number 1 right here between the sensory neuron and the interneuron. And then we have synapse number 2 between the interneuron and the motor neuron. Polysynaptic just means there's at least 2, and sometimes way more than 2, but at least 2 synapses. On my reflexes that are monosynaptic, that have 1 synapse, I'm actually missing one of my kinds of neurons. Which of these 3 neurons is usually missing when we have a monosynaptic reflex? Is it the sensory, the motor, or the interneuron? If I'm monosynaptic, who do we skip? Yeah, exactly. If you have a monosynaptic reflex, you have no interneurons. No interneurons. Because I only have 1 place where neurons talk to each other. If I only get to have 1 place where neurons talk to each other, what that means is I'm not going to process. There's going to be no processing. Every time I detect this sensory information, I will always respond the same way. So in a monosynaptic reflex, we have 1 synapse, or we just have 2 neurons. We're missing those interneurons. If we have an interneuron, we're looking at a polysynaptic reflex. The next way I'm asking you to classify reflexes is if they are cranial or spinal? Hopefully we can, let me see if I can phrase this question as easily as possible. How can you tell if a reflex is a cranial reflex or a spinal reflex? What would the picture have to show me if it's cranial or spinal? What am I looking for in my picture? Yeah, I love it. Tian's like, you would literally have part of the spinal cord, or you would see part of the spine, absolutely. Or you'd see part of the brain. Granted, we may not know what that part looks like, right? Because it just looks real sketchy. Basically, what we're looking for is a spinal cord. Do we or don't we see a spinal cord? So, not a trick question on this reflex here. Do we or don't we see a spinal cord in this reflex here? Do we see a spinal cord? Yes, we do. Yes, I love it. So since I see a spinal cord, which is all of this right here, right? I hope by this point in the semester, we know that that's the spinal cord, right? Since we see the spinal cord, we know that this is a spinal reflex. If you see anything else, it can look pictures in your lesson outline that show you what cranial reflexes look like. But the short answer is if you don't see the typical spinal cord, automatically we're saying that's a cranial reflex. One last thing I'm asking you to clarify or to classify with your reflexes, whether they are ipsilateral or contralateral. What do these words refer to? Ipsilateral and contralateral. What does this even mean? Yeah, I like it. So when we talk about ipsilateral and contralateral, big picture, we're looking at where that motor information goes out. If something is ipsilateral, it goes out the same side as the sensory information came in. If something is contralateral, it goes out the other side that information came in. So my reflex here, here's my sensory information, shown with this sensory neuron here in yellow. It's coming in on this side of the spinal cord. We synapse with our interneuron. My motor neuron goes out the same side of the spinal cord. This reflex is an example, of an ipsilateral reflex. We were all about to freak out a whole bunch when Dr. Allis started to circle contralateral. This is definitely an ipsilateral reflex. Okay, so I'm promising you on the exam, we're going to have something like this. It won't be this. It won't be any of the pictures from the lab packet. It's going to be different. I'm going to ask you to classify these reflexes. How do we feel right now about the prospect of getting a reflex we haven't seen before? Thumbs up, thumbs down, gritting our teeth. Kira's looking all cool with her sunglasses. Kaylin's mad at me. I know. Dr. Allis is a mean teacher. Yeah, so we're kind of on the fence. What I would say is with your studying, really the best way to study and prepare for this is just to remind yourself what each of these words mean and remind yourself how we saw them on the pictures of the reflexes that we did together. Remind yourself of what it looked like on this reflex that we worked on. So just keep remembering what the words mean. If we remember what they mean and how to apply them, I think that'll help us go through some of those emotions a little bit faster. Kaylin has a lot of emotions. I understand. It's all good. All right, let's do some more practice. This time, we're doing practice about the tracts. Now, these kinds of questions that we see here, this is some of the basic kind of stuff that we need to make sure we know about tracts. If we know this basic kind of stuff about tracts, that will help us out with the critical thinking questions that I also gave you about tracts. So when we're talking about tracts, remember that there are two kinds of tracts. We have ascending tracts, and their name tells us literally they're going up. And then we have descending tracts. Their name tells us that literally they're going down. Think of these tracts, again, they're kind of like highways. So information can either go really fast up or really fast down. This is the way that your body sends messages. If I am sending information up, if we're doing an ascending tract, when I send information up, am I sending up motor information or sensory information? What kind of information goes up? Yeah, exactly. If we're sending something up to the brain, presumably, the kind of information that I send up is sensory information. The brain is the place where we process. It's the place where we predict what we should be doing. Sensory information goes up to the brain for me to process it. Hey, motor information is what comes down from the brain. So I send up some information in my ascending tract. I process it in the gray matter of the brain, and then I send down my prediction, what I think I should be doing, using a motor tract, which is a descending tract. First thing we need to make sure that we know, because 100% of the time this is true, if information is going down, if we have a descending tract, that kind of information is motor information. The brain will always only send out motor directions, what to do. When we talk about sensory information is what your body collects and sends up to the brain for processing. So 100% of the time, ascending tracts have sensory information and descending tracts have motor information. Now, when we talk about these tracts, remember that the tracts have different numbers of neurons in them. So one of my types of tracts only has 2 neurons in it, and the names of those neurons, this will give it away a little bit, the names of my neurons that are found in a tract that only has 2, they're called an upper motor neuron and a lower motor neuron. When I talk about my tracts that have an upper motor neuron and a lower motor neuron, were those ascending or descending? If I have an upper motor neuron, type those out, and a lower motor neuron. Yeah, exactly. Their name says they're motor, right? And we just talked about how motor tracts are descending tracts. So my descending tracts that go down typically from the cerebrum, here we'll give one away here, my motor tracts that can start in the cerebrum, your thinking part of your brain, my upper motor neuron sends its information down to the lower motor neuron that'll go out and talk to your muscles, that'll get the work done. When we talk about ascending tracts usually have 3 neurons. We talked about one type that didn't have 3, but most of them have 3 neurons. So these 3 neurons are called first order neurons, second order neurons, and our last one, third order neurons. When we talked about a type of ascending tract called the spinocerebellar tract, that one only had 2 neurons, the spinocerebellar tract. It was actually missing this third order neuron. Do you remember where the third order neuron started or where it was going to in the brain? Yeah, exactly. So my third order neuron starts in the thalamus. The thalamus, that's the duck face in the middle of the brain. Remember when we're looking at that brain model? The third order neuron in an ascending tract is the neuron that's going to take the sensory information out to the particular part of the brain where it can be processed. If my second order neuron doesn't go to the thalamus, so in the case of the spinocerebellar tract, the second order neuron never made it to the thalamus. So if I don't make it to the thalamus, I don't have a third order neuron. But pretty much every ascending tract, except that one special one we looked at, they're always going to have 3 neurons. The first order neuron that starts out in the spinal nerves, or it starts out in the body where I collect sensory information. That first order neuron goes synapses with a neuron in the spinal cord, the second order neuron. And remember, the second order neuron says, this sensory stuff is above my pay grade. We're shooting it up. So the second order neuron is what forms the ascending tract, goes all the way up to the thalamus, where it meets with the third order neuron that sends that information where it's supposed to go. So ascending tracts have 3 neurons. Descending tracts have 2 neurons. Descending tracts that are sending information down, they can start in the cerebrum or they can start in the brainstem. So the brainstem, that's going to be places like the pons or the medulla oblongata. Those places can decide what movements we're doing too. So as long as we're sending information down, either from the cerebrum or the brainstem, that's called a descending tract. If information's going up, like because it started in the spinal nerves, that's an ascending tract. Hey, by the way, here's kind of an application type question. Say I made the decision that I want to pick up my coffee cup and take a drink. I decided what I wanted to do. Would I be using a descending tract that came from the cerebrum or a descending tract that came from the brainstem? What do you think? If I consciously decided what movement I wanted to do, would those directions come from the cerebrum or the brainstem? Several of us are chiming in. If you decide to do a movement, so if we're talking about skeletal muscle where you're deciding what to do, that's a descending tract that started in the cerebrum. We have a name for those. If you remember, we called those direct motor pathways. Direct motor pathways, the ones that start in the cerebrum and you consciously control them. If we start in the brainstem, so in the pons, in the medulla oblongata, this would be what we call an indirect motor pathway. Indirect motor pathways, these are going to be unconscious. So these are going to be movements that you didn't necessarily decide to do, they just happened. So this is the kind of stuff, for example, that helps you stay balanced. That kind of information comes through an indirect motor pathway. You don't think about it when you start to fall over, or you don't think exactly how to not fall over. That kind of information comes from the brainstem, or comes from the cerebellum that's right next door to the brainstem. One last question about tracts. This question asks, which kind of tracts are found in the white matter of the spinal cord? Which kind of tracts are in the white matter? Yeah, exactly. Here's Dr. Hollis's tricky question, right? It's not ascending or descending. It is both ascending and descending. Reflexes, all about the gray matter of the brain, or the spinal cord, excuse me. Tracts, all about the white matter of the spinal cord. And so remember, for the sake of the lab exam, anywhere that we're pointing that's in the white matter of the spinal cord, Remember that my other fancy name for tracts is funiculi. So anywhere on a model that I'm pointing at white matter, that's called the funiculi, the tracts of the spinal cord. So we looked at this picture very briefly in class last time, and I told you how this shows you the general locations of each kind of tract. Over here on the right side of looking at all of the ascending tracts, all of the sensory tracts. On this side of the image, we're looking at all of the motor tracts. But remember, I told you how, imagine that we did a little bit of art here. All that stuff we see on the other side over there, we would see it on this side too. So if you folded over this paper with that wet paint, you'd end up with all of the white matter of the spinal cord filled with tracts. In particular, there's a couple of tracts I was asking you about. These tracts are the anterior spinocerebellar, so there's that tract we were talking about, and the posterior spinocerebellar. For us to be able to figure out, here's our options here, 4A and 4B. These are my two spinocerebellar tracts. For us to figure out who is who, we need to figure out which side is anterior and which side is posterior. When we're looking at our spinal cord, is the top or the bottom the anterior side? The top or the bottom? Who's anterior? Yeah, exactly. So the anterior is on the bottom side of this picture, anterior here on the bottom, which means that posterior is up here on the top. Hey, help me out in the chat. How could I tell from this picture that this was the anterior side and this was the posterior side? Any ideas? Okay, so Kira said one of the things that helped her was seeing the fissure. Yeah, there is more open space on the front side of the spinal cord. Yeah, that's a way that would work. Yeah, so the way that Fanshin mentioned is probably the way I would use when I'm thinking about it. See how these horns back here go much closer to the edge of the spinal cord than these horns up here do. There's more space in between. So those dorsal horns are a lot closer to the edge of the spinal cord than the ventral horns are. So the way I would use when I'm looking at it, I would say, oh yeah, these ones are really close to the part where they come into the spinal cord. This is my back side. This is my dorsal side. So this is posterior back here, or dorsal. This is anterior up here, or ventral. So when I'm looking at my two options, A and B, which one of these would we say is the anterior spinocerebellar? A or B? Who's anterior? Yeah, exactly. Once we figure out front from back, it's not a trick question, right? We're just using those directional terms. So anterior, this one's closer to the front. Posterior, this one's closer to the back, posterior. So 4A is my posterior spinocerebellar tract. 4B is my anterior spinocerebellar tract. How do we feel about that? Thumbs up, thumbs down. What do we think? Anterior and posterior. Pretty straightforward once we know our sides. Awesome. Okay. So let's do this one then. This is another application of our tracts. This time, instead of giving you a picture, this time I've given you the name of a tract. And I've asked you to make some predictions about that tract based on its name. So this is a tract called the vestibulospinal tract. The vestibulospinal tract. When I look at this tract, there are two parts to its name. The vestibulo part and the spinal part. When we look at these two parts of the name, does this vestibulo part, does this tell me where the tract starts or where the tract ends? This first part of its name, is this where it starts or where it ends? Yeah, exactly. This is going to be where my tract starts. So the spinal part then is going to be where my tract ends. Now, I don't need to know what this vestibulo part refers to, simply because I know what this spinal part means. What does this spinal part mean here in vestibulospinal? Yeah, exactly. Fanchin mentioned the spine or the spinal cord. This tract ends in the spinal cord, which means that this vestibulo part must be higher because the spinal part is always lower. This is a tract that information is going down. This is a descending tract. Okay, so my information is going down. Does that mean that I'm sensory or motor if my information goes down? Yeah, exactly. Remember that descending tracts are motor tracts. I'm sending that information, that's motor information, down. So let's skip ahead then. If we know that the vestibulospinal tract is a motor tract, which of these kinds of neurons is it going to have if it's a descending tract or a motor tract? 5 different types of neurons that I would have in tracts. Who am I going to find in vestibulospinal? Yeah, exactly. These lower and upper motor neurons. Lower and upper motor neurons. Remember, my wink, wink, nudge, nudge. First order, second order, third order, these are only in sensory tracts. Sensory tracts. Or the other word for them is the ascending tracts. You should never tell me that I've got lower motor neurons and upper motor neurons in an ascending tract. These guys only send information down. Just like you should never tell me that these first order, second order, third order neurons are sending motor directions. You should never tell me that either. So make sure we know the tracts they live in. And hey, check this out. We kind of already circled it, so this should help us. If the vestibulospinal tract is a descending tract, or it's a motor tract, how many neurons do descending tracts have in them, or motor tracts? How many neurons do they have? Yeah, so if we look at our options down here, if we're in an ascending tract, we would have 3. Descending tracts that have motor neurons only have 2. Descending tracts always have 2 neurons. Ascending tracts always have 3, except for that one that we talked about. Hey, I know that one of my groups had mentioned this before. This would be some good stuff for flashcards. So how I could see flashcards helping would be on the front side of a flashcard, what kind of information does an ascending tract carry? Back side of the card, sensory information. New card. Front side says how many neurons are in an ascending tract. Back side says 3 neurons. Front side of a new card. What are the names of the neurons in an ascending tract? Back side has these 3 names with their locations listed. So this would be some information that I think could be helpful to put on flashcards, because it's little distinct facts that we just need to make sure we understand. Because if we can remember that a motor tract, which is what this was, is a descending tract. And we can piece that together with how many neurons we have and what they're called. You can answer all of these questions just by knowing those little bits of information. So I'm checking out Fanshin's question. Yeah, so Fanshin brought up a good point that when we're learning these tracts, especially the ascending tract, in some ways it is kind of similar to a reflex. So there are 3 neurons involved. I would say that our second order neuron doesn't really process it in the same way that an interneuron and a reflex does. But our second order neuron does process it enough to know that it can't do anything with it, and it sends it straight up to the brain. So when you're thinking about it, it's similar in some ways to reflex arc. But remember that these tracts are all about the white matter, and my reflexes were all about the gray matter. All right, we just reviewed a lot of information. How are we feeling at the moment? Or what questions do we still have about this? Okay. I'm getting several thumbs up, so that's perfect. And you can always come back to the video and practice these kinds of questions just to get yourself thinking these ways. So our big hint when we looked at this question at the beginning was to split it up into 2 parts, split the name into 2 parts, because once we did that, we could predict all of these other answers to these questions. So anytime you see a big long name of a tract, we're looking for, where do I have spinal? Or sometimes they're called spino if they're an ascending tract. Where's that spinal part? Is it at the beginning or is it at the end? If you can tell that, that'll help you to know if we are ascending or descending. And as long as you memorize these facts, you can totally make those connections as well. All right, we are going to jump back in to lesson number 12 where we left off last time. I believe the last thing we talked about was actually the vestibulospinal tract. Check that out. Tie into our group work, right? So the last thing we talked about was the vestibulospinal tract. This was getting us ready to talk about ALS. So ALS, let me type its full name here, amyotrophic lateral sclerosis, ALS. Most of us have probably heard of ALS, or it's sometimes called Lou Gehrig's disease, because of the recent ice bucket challenge. So it wasn't that recent now, I guess. I'm feeling a little old, but a few years ago, there was a challenge going around on Facebook and the like, where famous people and normal people alike dumped buckets of water, cold water on their head, and donated some money for ALS research. So this endeavor to raise money for ALS was a great way to bring awareness to this disease. Let's talk about how it works. Normal situation, you're looking at a normal muscle here, and you're looking at a normal neuron that's sending messages or sending impulses to this muscle tissue, causing this muscle tissue to contract. In patients who have ALS, a couple of things happen. So the big word for you to know with ALS is atrophy. When we use the word atrophy, it basically means that things get smaller. Atrophy. So in ALS, the things that get smaller are both our muscle tissue and our neurons. Neurons are what send the messages to muscles for muscles to contract. If muscles don't get a message to contract, they have no reason to contract. And the longer we go without contracting, the smaller those muscles become. It's kind of like that use it or lose it idea. It's very true with muscle tissue. So use your muscles or you're going to lose those muscles. Now, the thing with ALS is scientists were trying to figure out, is it that the muscles waste away first, which causes the neurons to stop talking to them? Or is it because the neurons stop talking to them that they're wasting away? Scientists wanted to figure out exactly why these patients lose all of their muscle tone. What ultimately typically ends up happening with ALS is the patients end up losing muscle tone to their diaphragm. And when their diaphragm is no longer able to contract, they're no longer able to breathe. So it's very important that ALS be identified as early as possible, try to treat it to prevent diaphragm wasting. How many of you in the chat, help me out here, how many of you have had a chance to read this thing before class today? Okay, so a few of us have. Awesome. I'm realizing right now as I look at this that my picture up here does not have underlining. Let me pull up my outline so I can underline it for my friends here on the recording. Can you tell me what page we're on in the packet? What page is this? Page 10. Perfect. Okay. So let me add my underlines here so that we are all on the same page. All right, here we go. So I ask you about right here and right here. And finally, down here. Now, if we were in class together, I would pull up my my little thing where you text stuff into me to tell me to answer some questions here. But we've got the chat going on here in the side, so we can just do that here in the chat. Let me tell you what we're looking at, and then we'll work through it together. This is something called a scientific abstract. So an abstract is where a scientist summarizes something that probably took them like 2 or 3 years of research. They put it all together in like one paragraph. So this is all of the most important information in this person's research, summarized down to one paragraph, a scientific abstract. Now, this is going to make me feel really old, but I'm curious. This paper came out in 1991, so this is not the most cutting edge research in ALS, but I'm curious how many of you, this paper is older than you are. Is there anyone in class today that is younger than this paper? Yeah, it's older than Kira. Kaylin's like, yeah, I wish. Yeah, me too. I'm not as young as I once was. Okay. So not too many of us. Usually when I ask a class, like half of my class when we're in person is like, oh yeah, I'm younger than this paper. I'm like, oh, I'm so old. So this is research that is older than some of us taking the class right now, but it's kind of groundbreaking in the sense that the goal of this paper was to figure out in our motor pathways, we know that they're not working correctly. We know that our muscles are not getting a message to contract. They wanted to figure out, these scientists were trying to figure out if we were having an issue with our cortical neurons or our lower motor neurons causing these muscles to not contract. Now, we could rename the word, they use cortical neurons. We called those upper motor neurons. That's what we called cortical neurons, upper motor neurons. ALS, Lou Gehrig's disease, is an issue with my kind of tract in the spinal cord that has upper motor neurons and lower motor neurons? Was it ascending or descending tracts that have upper motor neurons and lower motor neurons? Yeah, exactly. So I'm going to put a little note for us here at the top. ALS is an issue with descending tracts. My tracts that send out motor information. These are the kind of tracts that have upper motor neurons and lower motor neurons. If my descending tracts aren't working, I'm not going to cause my muscle to contract, and my muscle is going to waste away. Okay, so let's look at this abstract and see if we can decipher what these scientists found out. So we're talking about ALS, so amyotrophic lateral sclerosis. We are losing some neurons. We need to figure out which neurons they are. That's what my first sets of sentences mean. Here we go. This is what we're looking at here. So they took a cross-sectional area of pyramidal cells in layer 5 of the foot and tongue areas of the precentral gyrus. Ooh, that is scientific sounding, right? Here's what I want you to do for me though. Can you tell me in the chat, I've underlined these areas in this yellow color here. Can you tell me in the chat what of those words we do understand? We may not know what a bunch of the words mean or what that statement means. What are some of the words that you recognize in this statement here? Okay, I like it. So we are recognizing this right here, precentral gyrus. Thank you, Tien. It always makes me nervous when I do this in class with students and they don't send me foot and tongue. And I'm like, dude, y'all, if we are this far in the semester and you don't know what your foot is, I'm in trouble. So, yep, so we know the foot and the tongue. Yeah, and so then we talk about cross sections, so we're cutting things. Okay, so let's put this into things that we understand here. When we talk about the precentral gyrus, Audrey mentioned that's where the motor homunculus is. When we're talking about the precentral gyrus, is that going to be the location of upper motor neurons or lower motor neurons? Who would live in the precentral gyrus? Yeah, exactly. The precentral gyrus is where upper motor neurons live. So when we talk about what this statement in yellow means, the statement in yellow is talking about they're looking at upper motor neurons. Looking at upper motor neurons. Notice, by the way, that they're checking this in 12 cases of what they call classical sporadic ALS. Hey, technically in your packet, this classical sporadic form of ALS is also underlined. And I like to point out that there's this word sporadic in here. When we were talking about Creutzfeldt-Jakob's disease, what did sporadic mean? Do you remember if someone has sporadic Creutzfeldt-Jakob's disease? Yeah, exactly. Sporadic Creutzfeldt-Jakob's disease, we don't know where it came from. It just happened randomly, basically. So what this is telling us is 12 people who had no family history of ALS. They don't have genes that code for ALS. These were the patients that they studied. So they took a cross-sectional area, meaning they took a little slice out of the places on their precentral gyrus that corresponded with the foot and corresponded with the tongue. So if you go back to your lesson 11 packet, you could look at the motor homunculus, the space that was for the foot and the space that was for the tongue. They took a little slice of the cells that lived there. They wanted to see how big the upper motor neurons was. That was the first part of their study. Then they looked at the areas of motor neurons in the hypoglossal nuclei and in the ventral horns of the L4 segment of the spinal cord. Let's do like we did before. What are some of the things that we understand in this green area? What are some of the words that look familiar to us? Okay, yep, so we've got spinal cord. That's our current chapter, right? Yep, the ventral horn, we know about that. Yeah, so I noticed a couple of us mentioned hypoglossal. Why do we know hypoglossal? Do we remember why we have heard that before? Yeah, exactly. So this is this thing called the hypoglossal nucleus. This is the place where the hypoglossal nerve connects to the brainstem. The hypoglossal nerve, cranial nerve number 12, is the cranial nerve that's responsible for moving the tongue. So we are looking at the place where the neurons that help to move the tongue, the hypoglossal nucleus, and the neurons that are in the ventral horn of the spinal cord are located. The hypoglossal nucleus is where the lower motor neurons going to the tongue, where we would find those. This is the lower motor neurons for the tongue. And the ventral horn of this part of the spinal cord, so down by lumbar vertebra number 4, down at the very bottom of the spinal cord, this is where we would find the lower motor neurons that go to the foot. So when we look at our green section here, our green section is looking at lower motor neurons. The goal of this study was a chicken or an egg type thing. Is it that we aren't sending the messages in the brain to make contraction happen? Or is it my neurons that synapse with the muscle tissue that aren't getting the message, that aren't sending the message? We looked at both of these types of neurons to figure out which ones were smaller. Who's dying first, basically? Is it the ones in the brain or is it the ones in the spinal cord? Who's not working? Well, my blue statement tells us who's not working. Cortical neurons and lower motor neurons were significantly smaller in the cases of ALS than in the controls. Can we summarize this in easy words using what we talked about over here? What did they find about which kind of motor neurons was the problem? I like it, Kira. I don't know if you purposely made it sound a little thug, but yeah, they both bad. Yep. So when we're talking about ALS, we were trying to figure out, is it the upper motor neurons or the lower motor neurons? Well, what they found is that it's both neurons. Both types of neurons, the upper motor neuron and the lower motor neuron. So both types of motor neurons, I'll use our fancy word, they've atrophied. Both types of motor neurons have gotten smaller. So let's review what this paper showed. They started by looking at the places where the upper motor neurons live. So they live in different places in the precentral gyrus, the place where you decide what movements you're going to do. They looked there for the upper motor neurons, and they looked in either the brainstem, the place where the hypoglossal nerve connects, or in the spinal cord to find those lower motor neurons. They measured the size of the cell. And when they measured those sizes, they found that both of those types of neurons had gotten smaller. So this paper didn't solve for us the chicken or the egg mystery. Is it an issue at the top or is it an issue down lower? Well, we don't know for sure. We just know that both of them are malfunctioning. And because both of them are malfunctioning, we are not contracting our muscles correctly. As you are working on the lesson number 12 assignment, it's going to ask you about ALS. This is your reference for what we're learning about ALS. Make sure we know that this is a problem with those descending tracts or a problem with our motor tracts. Make sure that we know that it's both the upper motor neurons and the lower motor neurons that are having issues in ALS. Thumbs up on this abstract or any questions? Oh, good. Yeah, Kaylin says it's not too bad. When you look at it and you figure out what words you actually understand, it makes it a lot less intimidating, right? Scientific articles can be written real highfalutin, but focus on what you know and you're good. All right. Let's talk about something that's going to directly relate to lab. So this is an image that should remind you of one of the models you're labeling for the spinal cord. Let's see if we can remember. I'm looking at a spinal cord here. I've got a vertebra around it. Is this, which kind of vertebra is this? Cervical, thoracic, lumbar? Which kind am I looking at in this picture here? Yeah, exactly. This is one of my cervical vertebrae. Remember, there were two things that tell us that. First thing is right here. This is what we call the bifid spinous process. Bifid spinous process, meaning it's got two parts that branch off of it. All of my other spinous processes just have one. So a bifid spinous process, that's cervical vertebrae. The other thing that tells us it's a cervical vertebrae is right here. These two little holes called the transverse foramen, These are holes for blood vessels that go up to the brain. So the bifid spinous process, this is a cervical vertebra. Now that's lab stuff. What lecture stuff are these meninges. We want to make sure we remember where each of the layers are found. And we also want to make sure that we know about our spaces. So when we talk about the spaces around the spinal cord, they're in between these meningeal layers. So on the very outside of the spinal cord, the first space for us to be familiar with, that's the epidural space. The epidural space. Epi means above. Dural means the dura mater. The epidural space is on the outside of the thick dura mater out here. Hey, the epidural space is the place where epidural anesthesia goes when a woman is giving birth. Do we remember from lab, or can we tell from the picture, what fills this epidural space? What's in here? Yeah, exactly. So out here, this is the stuff I told you in lab. It's kind of like the bubble wrap around the spinal cord. We've got a bunch of adipose tissue out here in the epidural space. And check this out. The most effective kinds of epidural anesthesia are the kinds that match the chemistry of the stuff out here. Let's review way back to unit number 1. What are some of those words we use to describe things like adipose tissue or fat? What's some of our chemistry words? Exactly. Hey, this is great review for our final, right? So when we talk about adipose tissue, or when we talk about lipids, which is what fills adipose tissue, that's things that are nonpolar, that's things that are hydrophobic or lipid soluble. The most effective epidural anesthesia medications are medications that are nonpolar, that are hydrophobic, that are lipid soluble. Lipid soluble. So if we know what's out here, we can do that chemistry stuff, all those things we talked about before. The epidural space filled with adipose, this is where we inject epidural anesthesia. The other space for us to know is the subarachnoid space. The subarachnoid space. This one is underneath the arachnoid mater. This is filled with cerebrospinal fluid. So the cerebrospinal fluid that is encasing the spinal cord is found in a place called the subarachnoid space. Now, I want you to please make a note for yourself on your outline, so on your picture here. Next to this place called the subdural space, let's put a note that it's not there unless you're dead. The subdural space is not actually present until the time of death. At the time of death, the dura mater will separate from the arachnoid mater, but it's not actually there. It's not a fluid-filled space until you die and things shrink. So I tell you that to say I will use this as a distractor answer on homework assignments or on the exam. The subdural space is not real. The real spaces we're talking about, the epidural space, where we've got adipose tissue, the subarachnoid space, where we've got cerebrospinal fluid. Hey, there is actually one other place in the spinal cord where we have cerebrospinal fluid. Yeah, exactly. Like Kira mentioned, we do also have some cerebrospinal fluid in here in the middle. So the central canal, the other place in the spinal cord where I have cerebrospinal fluid. Here in the subarachnoid space and in the central canal. Then we've got the adipose tissue on the outside of the spinal cord. Make sure we know the epidural space and the subarachnoid space. So one of the reasons we're learning about these spaces is to help us think about a procedure called a spinal tap, or they're sometimes called a lumbar puncture. In a lumbar puncture or a spinal tap, we're taking a sample of cerebrospinal fluid. We just talked about the different kinds of things that we find in the spinal cord, whether it's adipose or cerebrospinal fluid or all that stuff. If we're doing a spinal tap where we're trying to collect cerebrospinal fluid, where are we probably going to try to take that fluid from? Which of these spaces, what letter do we think we would use to collect that fluid? Yeah, several of us have voted correctly. The place in the spinal that we would collect cerebrospinal fluid from is the subarachnoid space. That's where our needle is going to go for a lumbar puncture or for a spinal tap. Usually when we do this question in class, I'll get some answers that say the central canal too. The central canal does have cerebrospinal fluid, but I can't get to it. So there is cerebrospinal fluid here, but I wouldn't sample from there. That would damage my spinal cord. I'm going to sample from the subarachnoid space on the outside of the spinal cord. I have to be careful always when I'm doing a spinal tap to make sure that I don't damage my spinal cord. But I can actually, I pick where I'd like to sample from to help us out to make sure that we don't bump into the spinal cord. I just blocked it. Shoot, hopefully you have your notes in front of you. I just popped up a poll for you. Do me a favor and vote in that poll. I want to know where we would do a spinal tap. So I'm going to read you the answers. I'm going to hide the poll and read the answers to you, and you can put your vote in there. So answer choice A is saying we would do a spinal tap at the level of C1 and C2, so at the very top of the spinal cord. Second answer is T1 and T2, so in the thoracic region. Third answer, C, is L1 and L2. And D is L3 and L4. So C1, C2, that's A. T1, T2, that's B. L1, L2, that's C. And L3, L4, that's D. I got a few non-responses. Give it your best guess. It's completely anonymous. I don't care if you're wrong. 5, 4, 3, 2, 1. All right, let's see. A little bit divided. We are mainly divided between B and D. So let's look at our options. I'm going to close that poll so we can see it all together. B and D were our top picks. So we're hypothesizing either that we would do it between the thoracic 1 and thoracic 2 vertebrae, or we do it between L3 and L4. When we're trying to consider where we would do a spinal tap, our goal is to make sure that we don't bump into the spinal cord. If we want to make sure we don't bump into the spinal cord, we want to go to a place that should theoretically be below where the spinal cord ends. So the spinal cord, if you go way back to the beginning of lesson number 12, it ends around the level of L1 or L2. That's where we have that thing called the conus medullaris. If I want to do my very best to not damage the spinal cord, I'm going to do my spinal tap down here between the L3 and L4 vertebrae, because if I do it down that low, theoretically, the spinal cord should be done. It ended up here. I'm 1 to 2 vertebrae down below where it was. The only thing that will be in the canal would be my spinal nerves, those things called the cauda equina. So a lumbar puncture or a spinal tap, there's different names for it. We do those down at the level of L3 or L4, the goal being to collect the fluid around the spinal cord and not bump into the spinal cord. I see Audrey mentioned that she's had several of these before. That sounds scary to me, but I'm glad, Audrey, that you've made it through all of them safely. Kira asked a question about if you have extra lumbar vertebrae, where the spinal cord would end. That's a very interesting question. I don't know for sure. I would hypothesize that it might go down a little bit lower, just because the nerves that go out in between each of the vertebrae help to determine the length of the spinal cord. But yeah, that's a very interesting question. If you ever get a MRI done, Kira, and you find out where your spinal cord ends, I would love to know. It's a great question. All right, so when we're talking about doing a spinal tap, we're doing it down low, L3, L4. That's the goal. And remember, we're going into the subarachnoid space. So notice we're doing a spinal puncture or a lumbar puncture spinal tap, going here into that subarachnoid space where there's cerebrospinal fluid. Notice that the epidural anesthesia goes into the epidural space. So an epidural should not go through any of the layers of the dura mater. We should be going into the space that's around the spinal cord. Epidural space for epidural anesthesia, and into the subarachnoid space for a lumbar puncture or a spinal tap. Thumbs up, thumbs down on epidurals and spinal taps. What do we think? I especially hope that we remember where an epidural goes, since it's literally its name, right? I really hope we remember that one. All right, let's talk about the disorders we have that involve the spinal column and can actually involve these meninges as well. When we talk about the importance of the vertebral column developing correctly, we are talking about a disorder called spina bifida. Spina bifida. So spina bifida is something that doctors screen for all the time. I remember when I was in middle school, when I was getting a physical done, they have everyone bend over and they feel all of their vertebrae. Spina bifida can exist in a form that you wouldn't even know that you had it unless the doctor noticed it for you. So spina bifida just means that something happened with our spine. It didn't form correctly. Over here on the very far left, we can see what it normally looks like. You can see the spinous processes of each of these vertebrae. So the spinous processes of the vertebrae, notice that the meningeal layers, all the meninges, inside the vertebral column. They're plenty safe and sound. Sometimes everything forms correctly except for one spinous process on one of our vertebrae, typically a lumbar vertebra. If this is the case, you probably won't notice anything wrong in the functioning of your back. Your spinal cord is going to be just fine. You might notice a little tuft of hair on the lower back in the place where the vertebra didn't form a complete spinous process. This is the form, the least severe form of spina bifida, called spina bifida occulta. Spina bifida occulta. Basically, spina bifida occulta, it means you don't really notice it. The reason there's this little tuft of hair is because early in embryonic development, you're a flat sheet of cells. That flat sheet of cells kind of folds over and makes something called the neural tube. That neural tube is going to turn into the vertebral column. But if we don't quite fuse all the way, if we don't quite close that neural tube, you end up with some of the cells that either would have become nervous tissue or they would have become hair. Well, they end up becoming hair in the place where they didn't become nervous tissue. So spina bifida occulta, the two ways you'd know you have it is either there'd be a little tuft of hair where the vertebrae didn't fuse, or you might feel the lack of that spinous process. Just like Kaylin mentioned, this is the mildest form. Occulta means hidden, so you may not even know you have it because it's not debilitating in any way. With my other forms of spina bifida, you definitely know that you have them, and you know it at the time of birth. So let's talk about the next form of spina bifida, and we jump up a bunch on our severity scale here. So my next form is what we see right here. This is what we call meningoceal. Meningoceal. So in meningoceal, this is a form of spina bifida where only the meninges leave the spinal cord. So notice I still don't have a complete spinous process. I've got a hole in my vertebral column, but my meninges decide to go out and fill that hole, if you will. Over here in spina bifida occulta, I just grow some extra connective tissue. It's no big deal. Over here, my meninges decide that it would be fun to extend out. So I've got cerebrospinal fluid that's extending out here. I can see this bulging area here where cerebrospinal fluid is right underneath the skin. Meningoceal and my more serious one we're going to talk about here in just a moment, both of these are things that would be treated right away. We need to, because think about this. If something were to happen to this skin and we were to rupture it, for example, and you'd get a cut, this is literally a direct line to the nervous system. So babies, this would be corrected either right after birth, or if it's very severe, potentially in utero. This is cerebrospinal fluid inside the meninges here in this area. So meningoceal, I've got this fluid-filled space made of just the meninges. Very serious. But this is the most serious version that we see over here. This is called myelomeningocele. Myelomeningocele. This myelo part looks like myelin, myelomeningeal seal. Not only are we missing the spinous process, not only did the meninges and the fluid go out here, but so did the spinal cord. So in myelomeningeal seal, we've got myelin, or we've got nervous tissue that's gone out here into this fluid-filled sac. So the spinal cord itself is actually protruding out of the vertebral column into this fluid-filled space that we see on the back. This, by the way, spina bifida in general, this is why the CDC recommends that all women of childbearing age take a prenatal vitamin, whether they're planning on getting pregnant or not, because what makes sure that that flat layer of cells forms a complete neural tube is folic acid, one of the big important things in a prenatal vitamin. So low levels of folic acid can lead this neural plate, or neural tube, excuse me, when all your cells fold over on top of each other. If they don't quite close all the way, we might get a little tuft of hair, have no bones. If there's more of the gaping space, those meninges might come out. Or even more of the gaping space, the spinal cord itself might develop out there. So spina bifida occurrence goes down dramatically with those prenatal vitamins because of that folic acid. All forms of spina bifida, we've got issues with the vertebrae not forming correctly. And then meningeal seal and myelomeningeal seal. Those are both issues with those meninges. Let's talk about another disorder. We're doing the parade of disorders today. The next disorder we're going to talk about is something called shingles. So let me show you what shingles looks like here. Shingles is a very painful rash that you see. It can show up anywhere on the body. It's very common on the trunk, but again, it can extend either down into the arms, down into the legs. So the deal with shingles is that shingles is a rash that we see covering something called a dermatome. A dermatome. So this picture is showing you what dermatomes are. Dermatomes are the areas on your skin that all send their sensory information back to the spinal cord using the same spinal nerve. So for example, this area that I see right here, this line of skin on the front and then onto the back as well, that line of skin all sends its sensory information back to the spinal cord using the T5 spinal nerve, so the thoracic number 5 spinal nerve. Or if I look down a little bit lower, down by the belly button, all of this skin here in a line sends its sensory information back to the spinal cord using the 10th thoracic spinal nerve. So when we look at the thorax, when we look at the chest, dermatomes tend to be horizontal. We see them here going in a line. When we look down at the arms or we look down at the legs, dermatomes tend to run vertically. So all of this skin, so notice here from the shoulder down all along the back of the arm, all of this area sends information back to the spinal cord using cervical number 7. So the spinal nerve, that's C7. These dermatomes and the way that they're arranged matters because it helps you to predict where the shingles virus or where the shingles rash is going to be seen. So notice on this individual how they have a line of a rash. With shingles, it'll typically be on one half of the body. So this is this patient's left side of the body. Notice that they probably have a rash here across the T3 dermatome, for example. This person has a rash that's extending down along their buttocks region. So we're probably looking at an S1 dermatome where they've got a shingles rash. This poor child's got a shingles rash down the back side of his arm. So again, we're looking at like that C7 back here. Shingles happens when the varicella virus reactivates and gives you this rash. So let me put some notes here on our slide. Shingles is caused by varicella virus. I'm sure you all can have that. What other illness does varicella virus cause for us? Yeah, exactly. So this is the same thing that causes chickenpox. Chickenpox. So here's how it works. Person gets chickenpox. This virus replicates inside their body. You get the normal rash all over the body. Then you're done fighting the chickenpox virus. It flames out. But some of that virus, so virus hides in the dorsal root ganglia. Dorsal root ganglia. Remind me, what kind of neurons live in the dorsal root ganglia? Who lives in the dorsal root ganglia? Yeah, exactly. So this is the location where sensory neurons live, where sensory neurons live. Okay, so here's how this goes. You have the chickenpox virus, your body beats it, except for a few little viruses that go dormant. So dormant means it's not replicating, it's chilling out. You get a little bit older, or you get a little bit stressed, you're not sleeping enough, or Chris is mentioning that she had some damage. Something goes wrong with your immune system in a part of your body. This virus that was hiding out dormant can actually come back to life, if you will. So it reactivates. And when that happens, that virus starts to make copies of itself. And it sends those copies out your sensory nerves back to the skin. So shingles is a super, super painful condition, not only because of the rash that you see here, but also because the way that you get that rash is literally the virus is exploding your sensory neurons. That's the way that these viruses cause a rash, is they copy and copy and copy themselves inside your sensory neurons and then kill those sensory neurons to be released on the skin. I gotta have a moment on my my soapbox here. Shingles, when we're talking about it, I have some relatives, somewhat distant relatives, that are just sure that that shingles is caused by the chickenpox vaccine. They think that shingles is caused by the chickenpox vaccine. Can you all help me out? Why does that get under Dr. Ellis's skin? Literally, like shingles. Why is that not correct? I should have made that like a group work question, right? Fix this statement. Shingles is caused by the chickenpox vaccine. Can we put to words why that wouldn't be correct? Thinking about what causes shingles. Okay, yeah, so Fanchin's using anecdotal evidence. Her parents didn't get the shingles vaccine and they got shingles. Yeah, so the precursor for you to get shingles is you have to have chickenpox. So you have to have chickenpox. You have to have this active virus in your body. That virus has to go dormant and hide in your dorsal root ganglia. If you never have chickenpox, you can't get shingles. You could get chickenpox later in life and that would not be fun. But if you don't have chickenpox first, we don't have those viruses hanging out in your dorsal root ganglion. They cannot lead to shingles. So you got to have chickenpox first before you have shingles. To all my friends who are older than that article, right, who, like me, caught chickenpox, didn't get the chickenpox vaccine, caught chickenpox, they do have vaccines for shingles, and I would highly recommend that to avoid this very painful rash that can show up anywhere on your body. So think about that as all of us start aging, right? That the shingles vaccine is a good one to help your immune system be on the lookout for any viruses that are hanging out in those dorsal root ganglia. Yeah, I can't remember the exact age, Audrey. It might be 50 or it maybe could be a little younger. I'm not sure. I have not asked my doctor about it yet, but I plan to in the near future. I know that people I have a friend, for example, who caught shingles in her 20s. So it's not just, we like to think about it as like just an old person issue. No, if you're really stressed and your immune system is depressed, it can be an issue for anybody. So maybe if you lobby hard enough, your doctor would give you a shingles vaccine a little younger, right? All right, one last thing that I want to talk about, and that is paralysis. So when we talk about a person being paralyzed, there are 3 different paralysis or plegias that we can talk about. So the first one in your notes is what we call paraplegia. Paraplegia. When someone has paraplegia, can you help me out in the chat? What's going on with someone who has paraplegia? Yeah, so in paraplegia, it's just the legs paralyzed, so just lower limbs paralyzed. I'd say just, but it's still severe. In paraplegia, a person has lost the ability to control their lower limbs. Yeah, they're going to be confined to a wheelchair. They're not going to be able to walk or move those limbs. The next kind of paralysis that a person can have is called quadriplegia. And quadriplegia has quad in it, meaning 4. So quadriplegia, upper and lower limbs paralyzed. When we're talking about paraplegia or quadriplegia, this has to do with where in the spinal cord a person had damage. So what you need to be thinking about, and here's where we kind of tie it into lab, is when we talk about the lower limbs being paralyzed, think about how we talked about the lumbar plexus and the sacral plexus being found kind of in the very lowest part of the spinal cord. Remember that thing called the lumbar enlargement was where the spinal cord got big because all the neurons going to the legs came out. If we damage above the lumbar enlargement, which is anywhere in the spinal cord, basically, we would develop paraplegia. If we damage anywhere at or above goal enlargement, which is where all those neurons going out to the brachial plexus came from, if we damage anywhere around there, we also lose the use of our upper limbs as well. So for paraplegia and for quadriplegia, it's all about damaging the spinal cord. Think about the level that you're at and whether the neurons are going to the arms or the legs at that level. If we damage the spinal cord at one level, anything below it is not going to function anymore. I did ask you to consider, though, one other kind of plegia or one other paralysis, and that's called hemiplegia. And in hemiplegia, it's actually just 1/2 of the body that's paralyzed. So this is not actually a spinal cord issue. Yeah, this is actually an issue with the brain. This is an issue with the brain. Yeah, so one of the ways, like Audrey mentioned in the chat, that we can see a patient develop hemiplegia is after a stroke. So a stroke is when someone has a blood clot and we stop sending the oxygen and nutrients that we need to that side of the body. A stroke could cause hemiplegia, where someone loses the ability to use their left arm or their left leg, right arm, right leg. It could be because of a traumatic injury that damages one part of the brain. Let's see if we can remember this. We didn't talk too much about it, actually. If I have a stroke that affects the left side of my brain, which side of my body is going to be paralyzed? We didn't talk a lot about it. Yeah, exactly. You all know it, though. You're whizzes. When we talk about brain damage, the left side of your brain controls the right side of your body. The right side of your brain controls the left side of your body. So if someone has lost control of half of their body, you can make a prediction as to which side of their brain was damaged. It's the opposite side of their body that's no longer functioning. So 3 paralysis disorders: paraplegia, where it's the lower limbs paralyzed; quadriplegia, where it's all 4; and hemiplegia, where it's just one half of the body. Again, likely due to something like a stroke, something like a traumatic brain injury, those kinds of things. Y'all got me talking. You got me up on my soapbox. Class went much longer than I intended. I apologize. Any last minute questions before I shut off our recording? I'll mention too here in closing, please try, if at all possible, to get chapter 12, the assignments, wrapped up today or tomorrow. We really need to start working on lesson number 13 because that is a beast. So if you can, today or tomorrow, try to wrap up the spinal cord, even though it's not technically due till Friday. Let's try to get it done sooner than that so you can start working on special senses stuff. All right, so tomorrow's class going to be all about predicting muscle movements. Check out those activities, the origin and insertion activities, last part of every lab packet since the midterm exam. That's where we're going to focus our time tomorrow. On Wednesday, we are going to start special senses. So plan, if you can, to start looking over that stuff, or at least come to class on Wednesday, and we'll cover special senses together. So check those out to get us ready. There's a question in the chat about our lecture final. Our lecture final is opening 2 weeks from today. So that is the 10th, I believe, the 10th and the 11th is when that lecture final exam will be held. All right, I'm going to go ahead and end the recording. I will stick around in the chat to answer questions, but I hope to see you tomorrow either in class, or if you have specific questions too, I have student hours tomorrow. So maybe I will see you tomorrow at 10 o'clock. Otherwise, I see you at 1 o'clock for class. Have a great day.